Good afternoon, everybody. Welcome to today's Coffee Science Seminar. My name is Mike Gidley. I am the Director of the Centre for Nutrition and Food Sciences within Coffee, and it's my pleasure to be the host for this presentation today. Before we start, I would like to acknowledge the traditional owners and their custodianship of the lands on which we meet today. I pay respect to their ancestors and their descendants, who continue cultural and spiritual connections to country. I recognise their valuable contributions to Australian and global society. So this seminar is scheduled to go till 1 p.m. At the end of the presentation, we will have a Q&A session. So if you have questions that you would like answered, please use the Q&A button, not the chat button. So the Q&A button at the bottom of the screen. And we will address them at the end of the seminar. So once again, please don't use the chat function. We'll only be looking at the Q&A section to get the questions for the presenter afterwards. So today's presenter, great privilege for me to present Dr. Bernadine Flanagan, a research fellow in the Centre for Nutrition and Food Sciences. Bernadine was the first employee of the Centre for Nutrition and Food Sciences in 2004, and has worked extensively with spectroscopic, particularly NMR and other analytical chemistry techniques to really understand the structures and their functional relevance of plant foods of various sorts, including ingredients and model systems. And at the moment, she's particularly interested in how these food structures change during digestion and fermentation and what implication that has for human health. So Bernadine is a UQ graduate, Bachelor of Science and a PhD in inorganic chemistry, and has been the co-author of 66 peer-reviewed articles, already over 3,000 citations. So clearly someone that people want to read, and also, as you will shortly find out, someone you would like to hear from. So please, Bernadine, take it away and tell us about dietary fibre structures and how they change during digestion and fermentation. Thank you.
Thank you so much, Mike, for that kind introduction. I too acknowledge the traditional owners and their custodianship of the lands on which we meet today and pay my respects to their ancestors and their descendants. So today I'm going to talk to you about dietary fibre. I really like it. I hope you like it too. You should, because dietary fibre is super good for you. So just a little bit about where you can find it. So dietary fibre is at the bottom of this healthy eating pyramid. So grains, vegetables and legumes are full of dietary fibre. It's great for improving laxation. It increases stool bulking. It can reduce the circulating blood cholesterol, and it can decrease the amount of glucose spikes in glucose after eating. So what is it exactly? A useful definition is that it's the edible parts of plants, plant foods that aren't broken down in the body, so by the endogenous enzymes. And so it's not broken down in the stomach or the small intestine, and that can be carried through to the large intestine where it can be fermented. So all of these lovely foods at the bottom of this pyramid, so the grains, vegetables and legumes, they all have plant cell walls. And the plant cell walls are made of polysaccharides, cellulose, hemicellulose, pectin, and lignin. And there's also in there other fibres, other sources of fibre are oligosaccharides and even resistant starch. So the important thing about dietary fibre is what it does. So what it does, it's available to be fermented by bacteria in your large intestine and to a lesser extent in your small intestine. And you really need to know how fermentable it is and the rate at which it's going to be fermented, because that really affects how you feel when you eat it. So if you have rapidly fermented dietary fibres it's fermented in the cecum or proximal colon, and that means that you don't have it carrying through to the distal colon, so it can't be providing short-chain fatty acids, which are beneficial metabolites of fermentation. It can also mean that if you get a rapid production of gas, and this can cause discomfort and flatulence. So that's not desirable. But again, if it's too slowly fermented, that can be a problem too, because if it's not fermented at all, it's not providing any beneficial metabolites, and all it's doing is bulking the feces, which is good in itself, but it's not everything we want. So what we want is fibres that are going to be fermented steadily along the tract. So you might need a range of fibres or a single fibre that's going to be slowly fermented all along the tract. So we get slow gas production and good amounts of short-chain fatty acid all along to provide those health benefits. So a lot of people traditionally think that if something is soluble, it's going to be more fermentable, but that's not necessarily the case. So you can have something that is soluble but very hard to ferment, like alginate, and you can have something insoluble like starch, but it is relatively easily fermented. And so it all depends on packing and swelling. So cellulose, for example, in a wheat bran, it's very densely packed and it doesn't swell, whereas the exact same cellulose, which is still insoluble in a fruit cell wall, it's very swollen and it can be fermented more easily. So we focus on plant cell walls, which, as I said, are the major source of dietary fibre in plants. And we look at primary plant cell walls because that's the ones you mostly eat. Secondary cell walls are the tougher lignified cell walls. So we like bran, we like cereals, and cereal endosperm cell walls didn't traditionally have a model for them, and that was something, a paper we published, Ganendra, our student, was the lead author on that, where we've published a picture of what the cell wall looks like. We've got a cellulose scaffold and it's got lots of polysaccharides attached to it, so heteromanans, arabinans, and then we've got not adhered to them, but in the cell wall as well, but that can be removed. We have arabinoxylans, and the arabinoxylans are cross-linked to each other, and also we have beta-glucan. So depending on what type of cell wall, this is a wheat cell wall, it has more arabinoxylan, but if you were looking at oat cell walls, they have more beta-glucan. So the most important polysaccharides in the cereal grains are the arabinoxylan. So you've got a xylose background with pendant arabinoses, and they can be either, you can have one arabinose on each xylose or two. And depending on what your structure is, you need different enzymes to break down this polysaccharide. So you need different ones to cleave the xyloses and different ones to cleave the different arabinoses. The structure is very important. So when you look at beta-glucan and cellulose, they're exactly the same monosaccharides. You've just got glucose. That's it. So it's the same monosaccharide, but the linkages are different. So cellulose has 1,4 linkage, and that's it all the way through. Whereas when you look at beta-glucan, every now and then it's got a 1,3 linkage as well as the 1,4, and that makes a massive difference in terms of solubility. So we need to think about what sugars we have and what their structure is. When we look at the other type of primary cell walls, type 1 cell walls, these are the ones found in fruits and vegetables. So in fruits and vegetables, the big difference is there's a heap of pectin. And as you know, pectin, it can swell and it forms gel. So it's the gelling, it's what makes your jam gel if you're making jam. And we also have xyloglucan. And so it has a much more open structure than the other cell walls that you find in cereals. So the major components of that are cellulose, xyloglucan, which this time is a glucose backbone with xyloses, and then some galactose. And you have petramannans and pectins. Pectins can have lots of amazing structures, but the one you see the most often is polygalacturonic acid. So it's just a heap of galacturonic acids, and they can be methylated and acetylated. So when you're analyzing for pectin, you can just see how much galacturonic acid you have, and that will tell you usually how much pectin you have. So we look at cell wall components, and we use different techniques to do that. So if you want to know what polysaccharides are in your cell wall, you need to know what monosaccharides you have. So we acid hydrolyze them and derivatize them and use either HPLC or GCMS. And the other thing we can do is quantify the pectin we have by hydrolyzing it to the individual sugars, derivatizing it, doing a colorimetric assay. So these two techniques were set up in our lab by Dr. Francesco Soni. For many polysaccharides, such as mixed linkage glucans, we can analyze them with megazyme assay kits. Megazyme assay kits are great also for looking at resistant starch or total dietary fiber. So if you just want to know how much of something you have, that's a good way to go. But we usually are looking for more than that. We don't just want to know the quantity, but we also want to know something about the structure. So that's when we use solution state NMR. So the samples need to be dissolved in a deuterated solvent. We can use a small amount of sample. It doesn't take very long for most of the samples to run. It can be as little as 5 minutes, longer for more detailed information. And we run at high temperature, and it's quantitative. So we look at polysaccharides. This is a lovely spectrum of starch, one of my favorite things. And we can tell differences in the branching ratio. Also, you can get some information about the degree of polymerization. So we know a lot about the structure, but what we need to know is how do these things ferment, because that's what's going to determine how useful they are for you. So we assess their fermentability by looking at in vitro fermentation. We take as homogeneous a substrate as we can. We add some basal medium, bicarbonate solutions, phosphate solutions, and reducing agents. And then we add all-important fecal inoculant. So the fecal inoculum, we've often used porcine inoculum. It's quite good because the pigs will eat a defined diet without complaining, and they're pretty reliable in producing their feces in a timely manner. But we've also now moved on to using human inoculum. It's obviously, we rely on donors for this, and we have more experiments coming up. If anybody would like to be a donor, that would be, can get in contact with Barbara Williams, who is our queen of gas production. And with the human donors, they're obviously not on a defined diet, and it's often difficult to make sure you get sample on time every time, but it provides us with it's just a different system which provides different information, but it's quite valuable. And once we've inoculated the samples, we then start measuring the gas production. And this needs to be done regularly, if not constantly. And if we use we have a smaller number of bottles, we can use an automated gas recording system, which is lovely. If we don't, if we want to do a big number of bottles, then we do it manually, and we need to be here around the clock. We also like to do timed removals to assess changes in the substrate over time. And that requires you to be there, obviously, to take them off at particular times. So these gas experiments can go for up to 96 hours, and they're a lot of effort, but the results are generally worth it. So we look at changes in the structure, starting here with simple system. So we're going to start with the single polysaccharide and build all the way up to whole foods. So this is some work from our excellent PhD student, Guan Li. She started with the wheat and rye arabinoxylan and looked at the structure as it changes. So we've got different enzymes working to break down the polysaccharide, so it gets reduced to smaller pieces, shorter, and also then, and we also wanted to monitor whether the arabinoses would be cleaved first. But what we've found is there's very little difference in the arabinose to xylose ratio as time goes by, but you do mostly get shortening of the arabinoxylan, so through this silane bonds breaking. And we can monitor how it's degraded, and you can see by 8 hours all of the polysaccharide is gone. So once you've looked at the individual polysaccharides, you want to, and nice, simple, soluble things, wanted to make things a little bit. So we also, when you've done these fermentations, you need to look at other metabolites. So we look at gas, we look at the substrate changes, and we also look at the production of short chain fatty acids. And we can do this with solution state NMR. So it is quite a versatile technique, and we can look at everything in one go. We just take a sample from the top of the bottle, the fermentation bottle, and from that we can get short chain fatty acids. We can also see if there's any methanol production, which you see sometimes with pectins, and just everything else at the same time. So it's quite useful as a snapshot. As you can imagine, with these experiments, we end up with an awful lot of bottles. So the NMR is quite tedious if you need to do it manually, but fortunately over at the CAI, the Center for Advanced Imaging, they have a beautiful NMR instrument with a auto sampler, and you can run through 96 samples in a go. You don't have to be there, obviously, set it up, and you come back the next morning and all your lovely data is sitting there waiting for you. So you can also use that to measure all the substrates as well. So you just set up whichever experiment you want, short chain fatty acids or different substrates, and they can all run through. And it's a great way of looking at everything together. But if you want to look at something more challenging, so non-soluble, because as you know, the plant cell walls aren't all soluble, they've got cellulose in them, which is definitely not soluble. And it's very difficult to isolate cellulose from a plant cell wall, to isolate it cleanly and without changing its structure. So in our labs, we're lucky enough to have Dietary, and she set up this bacterial cellulose production method, and it makes these lovely pellicles. And if you grow the bacterial cellulose pellicles with another hemicellulose dissolved in the solution, you can get a composite. So these are our plant cell wall models. So we use solid state NMR to look at these composites, these cellulose composites, because solid state NMR can basically do anything. It can look at a range of samples. They can be solids, just dry, wet. You don't really need to do anything to the sample, but you need to pack it homogeneously. We can work with as little as 10 milligrams of sample, and we also use it to look at starch. And we use different techniques, but the one we most commonly use is this CP mass, and it looks at the rigid components in the sample. And it's takes 1 or 2 hours to run a sample. But you can see a lot of things from it. You can get the crystallinity of the cellulose. You can see the crystalline to non-crystalline ratios. And you can also see how the hemicelluloses are interacting with the cellulose. So if you get binding to the cellulose. And we have used these cellulose comments. So we know the structure, we know the interactions between the cell walls, between the components of the polysaccharides, and what we need to know then is how it ferments. So we've done in vitro fermentation of these cellulose composites, and this work was led by Shi Lu, our lovely PhD student who just graduated this year. So in this we had human feces from 6 generous volunteers, and we fermented the sample for 48 hours, and we took different time removals. So we start with we have xyloglucanol by itself as a pure polysaccharide, bacterial cellulose, and then a composite of bacterial cellulose and xyloglucan, and a mixture of bacterial cellulose and xyloglucan, and we also have wheat bran as a positive control. And what we look at is we see that there's no significant difference in the short chain fatty acid production. As we expect, the xyloglucan is fermented much faster than everything else and produces a lot more gas. And the bacterial cellulose and the xyloglucan initially fermented faster, and it's also to a greater extent than the composite. And we look at the degradation patterns of the VC XG composite and the mixture. And what we find that's interesting is that the xyloglucan is fermented faster in the BC XG composite than it is when it's by itself. So you get degradation occurring earlier. So although, and then we also see that the bacterial cellulose is fermented more when it's in the mixture. So the rate of the xyloglucan degradation is faster in the mixture, and the extent of the bacterial cellulose fermentation is greater in the mixture. And we use the solid state NMR to see the change in the bacterial cellulose composite, because you can't use solution state NMR or anything else to look at how the xyloglucan is disappearing. But here you can see it's going, it's being degraded before the cellulose. But the 56% cellulose degradation is quite a lot. So we wanted to compare how the composite degradation compares with the degradation of the onion cell walls. So just to up the complexity a bit, so we've gone from the bacterial cellulose composite to whole cell walls, which have xyloglucan, pectin, and cellulose. So to compare them, we have a triple mix of xyloglucan, pectin, and bacterial cellulose, and they're in the ratios that we would get from the regular, that we found from analyzing the onion cell wall. And we also have the pure polysaccharides, bacterial cellulose, and again, wheat bran as a control. So we found that each constituent in the mix was fermented to a similar extent as it was in when it's single. So the pectin in the triple mix is fermented to the same extent as it is in the when it's by itself, and you get comparable gas and short chain fatty acid production. Compared with the mix, the onion cell wall was degraded slower at the earlier stage of fermentation, which you can see here so the rate is lower at the start of the fermentation, but when you get to the end, you see that the extent of the fermentation has really picked up, and so it means that more extensive fermentation occurred at the end. So you have very slow initial degradation of the cellulose, but it's almost complete. So amazingly, you get 95% of the cellulose in the onion cell wall is degraded after 48 hours. And you can see that here in the solid state NMR. There's just nothing left, which is in contrast when you look at the composites, and very different from if you look at wheat cell wall, where the degradation is much lower. So those are just cell walls, but again, we need to up the stakes. So here we're going to look at something more realistic. So when you are eating food, you don't just have plant cell walls. You have whole cells, broken cells, and their cell contents. And one of the lovely cell contents is starch. So here we have a milled lentil, and you can see that many of the cells are broken and the starch is spilling out. But if you boil your kidney beans before you blend them, you can get all your starch encased within these cell walls. So if it's inside the cell wall, it's not going to be digested, and then it's resistant starch, and that means it can go through for fermentation. So starch can also be resistant, not just by being within a cell wall, but also because of its structuring. So if it has a high level of order, it's often more resistant to digestion. So we're looking here at starch granules, which is raw starch granules, but when you cook it, which you have to do because you don't want to eat raw starch granules, they're not very delicious, but when you cook it, you change the structure, so we lose all of this molecular order. But on cooling, the molecular order starts returning, and that's one of the processes of staling. So if you feel that the bread is stale, it can be from the reformation of these molecular order. But this can be resistant to digestion. That's another form of resistant starch, these cooked, cooled, and reordered starches. So we want to know the order of starches, and we do this with looking at solid state NMR. We can, you can also do it with XRD, but one of the benefits of looking at NMR is that you can do it in a very complex mixture. So we have here we have just nice clean starch. We make an amorphous standard by boiling starch and then rapidly precipitating it, or rapidly freezing it and then freeze drying it. And we compare the structure of the amorphous starch here, match them at this point, and then calculate the percentage of ordered and non-ordered. So we, working with Fred Warren, who was a postdoc with us for some time, I had 115 different starches, and I'd done the ordering, calculated the order of those for various reasons. So we had mutants, we had extruded starches, retrograded and amorphous, and also enzymatically digested. So I did the classic peak fitting, and using those results, Fred developed a predictive model. And so now when we want to look at the order of starch, all we need to do is put in the NMR into an Excel spreadsheet and use this little macro, and it pumps out the result within a minute. So you can find out the level of order. And it is very useful because we can use it in just complex foods, it works very well, even after you've got a sample that's gone through fermentation. So without any cleanup, you just take your starch straight from the digester, and you can look at the structure. So here we looked at the changes in structure during fermentation. This was published in 2018. So this is Fred's paper. And we found the molecular order before and after fermentation. And what we found is there's no significant difference in the amount of order in these starches, so that the ordered and the amorphous are being fermented at the same rate. So the only time you see the differences in this cooked and cooled potato starch and for this pasta. So generally they attack the crystalline and amorphous starch equally efficiently. So that's what starch does when it's out by itself, but what does it do when it's inside a cell? And some of this comes down to how well you chew your food. So if you have your starch still contained inside the cell wall and your other energy sources, so within the cell contents, the other thing we looked at is the other energy store. We have the starch in our legumes, but in nuts, we looked at lipids. So depending on how well you chew your food, it's going to determine whether your food is, the starch and the fats are digested, or whether they are able to be carried through for fermentation. So this is an published publication from Wild's group, and it looked at chewing almonds, so different sizes of almonds, and then chewing them. So it's looking at the bolus, so chew and spit, where you they measure the size of particles that would normally be swallowed. And people swallow quite large particles, so between 4 and 1 millimeters is the common swallowing size. And so we wanted to look at a range of sizes and see what the different fates are to the cell contents. So we started with in vitro digestion of legumes and nuts. So we blend the nuts, centrifusion when we wash them, and then we sieve them to get different sizes. So we can see what happens to the different sizes during the digestion and fermentation. So when we get our nice clean dietary fibre, as would go into the large intestine, we take that and do the in vitro fermentation. So just as before, we have have all the bottles, and then we have the substrate in the bottom, add the inoculum, and look at the fermentation. And we also do removal times. So you can see that the legumes are fermented they produce a lot more gas than the nuts, so they're fermented to a greater extent in this time period. So here we're only looking at a 48-hour time period. And you see that for the nuts, you get a larger amount of fermentation for the F, which is the smaller fraction. So this is the 250 to 500 fraction, same as the size for the legumes. And then we have clusters, which are more like the size that's commonly swallowed. So these are bigger cells. So we have less surface area and more cell contents. So when we look at the microscopy to see what's happening exactly to these samples, we see that the starch at the start in mung beans have tester, which the other legumes don't. That's just how they're processed. So here we have starch within the lovely cell walls. It's all nicely encapsulated. At 12 hours, it's still there. And this is backed up by the solid state NMR. We have this peak here, which is for the C1 of starch. But by 12 hours, 21 hours, that peak is gone ski, and there is no more, you can't see the starch anymore. And by 48 hours, all you're left with is the tester. When you look at macadamias, very interesting. The NMR is quite different because these are dominated by, no longer dominated by polysaccharides, but have a massive lipid peak. These are all normalized for the lipid. You can see at the start there is polysaccharide there, and in the microscopy you've got beautiful lipids encapsulated within the cells, and then as time goes on, the cells, you can see that there's still these broken cells, and they're still there at 12 hours. But by 21 hours, it's very hard to see any polysaccharide at all, and you can't see any of those like cell walls anymore. There's still something there, but not very much. By 48 hours, you can see that even the lipid droplets have changed. They have been emulsified and are much smaller. So there's a lot going on with the nuts, but often people think that the lipid is damaging for the bacteria, but what we find is that it's fermented well. So it could just be the delivery system that they're delivered with plant cell wall, so it's not a shock to the system initially. You've got structuring of with carbohydrates, and then you get degradation of the lipid over time when you've already got a good bacteria population going. The actual lipid profiling doesn't change much. We see the similar profile of triglycerides, polyunsaturates, and lipids over time. By 48 hours, it's just in the peanut and the small peanuts, it's just gone. When the cell walls are broken due to fermentation and the lipid is released, it doesn't adversely affect the activity of the resident microbiota in terms of the cumulative gas production. So I just want to summarize by saying that not all the fibres are created equal. You need to think about food processing. So that includes grinding, blending, cooking, and even how much you're chewing, because that's going to affect how much of your plant, how much of the energy you're going to get from the food, and how much energy is going to go through digestion, and how much is going to go through to feeding the microbiota. So, and this also will create differences in fermentation rates, even for down the line. So structuring is important, structure and architecture, and that's all affected by food processing. And all of these things affect the fermentation, fermentability, and the bacteria in your gastrointestinal tract. So I would like to acknowledge funding. We had a wonderful time in the AIC Centre of Excellence for Plant Cell Walls with a great group from University of Melbourne, University of Adelaide, and us. And also we've had this last section of work on macadamias and legumes is supported by Hort Innovation, Naturally Nutritious. I'd like to acknowledge my supervisor, Mike Gidley, for his wonderful support and advice, and all of our team members, past and present, who have thoroughly enjoyed working with. And I thank the people at the Centre for Advanced Imaging, Katya Starina, who looks after the solid state, and Greg Pierens, who looks after the 700 with the autosampler, and also the 500, because they just keep everything going. And without them, we couldn't do any anymore. So thank you very much for your time today.
Thank you so much, Bernadine. So please, everyone, have a think around some questions, put them into the Q&A, and we'll get to them. So Thanks to those who have already put questions in there. So I think that's a few places to start. So Tim O'Hare has been particularly prolific in his questions. So let's go to those. Some really interesting questions, Tim. So Bernardine, the first question is around whether there are any animal-based fibers that can substitute for plant-based fibers. So Tim uses the example of the traditional Inuit diet, which is essentially 100% animal products. So what does that do to their gut microflora?
Well, it would be very different. So fermenting, so that's basically proteins. So you go to get a lot more production of ammonia, less production of short-chain fatty acids. I can't imagine it's ideal. But there would also be, you probably get more fermentation of lipid as well. That's really, perhaps Mike would like to answer that.
No, I don't have an answer. I think what you just said at the end is particularly interesting. It is possible that maybe the lipids could substitute to a certain extent. I think there is an observation that though the Inuit diet is fine if you are not overeating, but if you have an excess of calories and you're on an animal-only diet, then that really does cause problems because you're now overloading particularly your microbiota with too much protein and lipid. The traditional Inuit diet was probably pretty calorie restricted, and so not so much of a problem. And second issue is in the environment they're in, then the need to ward off opportunistic pathogens wasn't so great because the bacteria don't survive that well in the climate either. So there's some arguments there, but it's a really interesting question because there are certainly, it is possible for humans to survive with no plant foods. It is possible, yes. A more sort of technical question, I guess, from Tim is around what happens if you deliberately add the products of fermentation of short-chain fatty acids. So he uses the example of the acetic acid that would be present in vinegars. You know, does that actually inhibit fermentation because you get product inhibition of fermentation?
So we have actually just done this. So we were looking at short-chain fatty acids. So seeing what happened if we put more short-chain fatty acids in and if they would be metabolized and what metabolites they would produce. Everything copes quite well. It didn't really do anything bad or good, but that would be different. So that in that case, we're delivering it straight to the large intestine model. But if you're eating it, it goes through digestion first. So I think it probably would never even see the bacteria.
Yeah, I agree. I mean, the acetate would be pretty rapidly taken up in the small intestine, so there's no real consequence in terms of the large intestinal fermentation from that point of view. So another question from Tim on the legumes that we'd use as an example there about amylase inhibitors and whether that has any impact. So in terms of the in vitro trials of digestion and fermentation, would you expect that to be different in vivo for when you do have these amylase inhibitors?
I think there is a lot going on there. I don't know if the amylase inhibitors are even as important as the dietary fibers, so they can soak up all your amylase as well. So there's a lot of things happening there yeah. So you've got the cell walls. You've got to get through the cell walls first. So you need to I don't know, the amylases are going to soak it up and everything else. So
yeah, I guess it's going to be a similar answer to the last one, isn't it? That there'll be an effect in the small intestine, but unlikely to have any effect
in large intestine.
In large intestine. Most of the amylase inhibitors are proteins, so they're also susceptible to either digestion or fermentation themselves. So that's another issue there as well. A couple of questions from Rob. Yeah, what's the combinations here? So what are the benefits or otherwise of a combination of fibre and acetate, and he used the example of vitamin B complex on the gut microbiome. So can you augment a good dietary fibre intake with other components, I guess is the underlying question.
Do you want to feel that? They have such different roles. I don't, again, so the vitamin B would be taken up earlier, and the short chain and the acetate too. So they're not really going to influence what's happening in the large intestine so much. And they just have other benefits. So yeah, so the more, if you're eating whole foods, you're going to get all of those things as well. So eat well, eat things that already have vitamin B and all the polyphenols, and then you shouldn't need to supplement as much.
Yeah, absolutely. So I think, as you said in the talk, then think of the plant foods and the cellular structures as a delivery system, so that you've got a way of targeting where you deliver these things to. Whereas if you're eating unprotected or purified materials, then they have the chance to be changed in the mouth and the stomach and absorb small intestine before they ever get to the large intestine. My personal view is, I think, a lot of these ingredients, unless they are really resistant to digestion, are largely going to have their effects before they ever see a substantial amount of gut microbiota. They might have effects on the microbes in the stomach, and there are microbes in the stomach, but unlikely to have an effect further down the track. Now, that is, a lot of people do swear on things like vinegar as being good for the gut microbiota. I haven't seen really strong evidence for that, and I would suggest it's probably better to have those things produced in situ by fermentation of carbohydrates rather than drinking them directly. But I think the jury has to be out on that. I'm not sure that the definitive experiments have been done. So guys, any more questions? Please don't be shy, put them in the Q&A, and we've got a little bit more time left, a few minutes if you would like another question answered. Okay. That's probably it, I guess. In terms of commenting, I guess we do hear the dietary advice all over the place around eating a varied diet rich in plant foods. That is the cornerstone of dietary advice globally. We have global consensus on that. And I think we need to be looking even more in this sort of work that Bernadine's been talking about, not in terms of what are the individual ingredients. So the dietary fiber don't think of it as an ingredient, it's a delivery system as well. So how do you capture that on a label? What is it? How do you describe the composition of something in terms of how it delivers rather than what it contains? And I think that's quite a challenge. It's a good measurement challenge, but it's quite a tricky thing to know how to do, particularly when you come down to how people chew their food differently, have different genetics and different detail in their digestive tract, different microflora, a lot of things there. I think we're seeing that the rather simple approach of saying you just look at all the ingredients in the food and you get an idea of how good it is for you. The sort of work that Bernadine has been doing and described today, that says there's got to be something else on top of that as well. That it's not just around the composition, that we've got to find a way of articulating what it is about the longer length scale structures that really cause that. You want to say anything more, Bernadine? Any other final comments before we let the good people go?
No, thank you. I'm very happy to.
All right. Okay. All right. So, is there another? Okay, one more question. John Dixon. What are the implications of insecticides and pesticide residues on food for microbiome function?
I really don't know. Again, small molecules, so I would hope that they'd be dealt with earlier. I can't imagine they do any good for you, but whether they would all make it to the microbiome, I would wonder.
I think that's right. So, and I guess if they do, then actually the microbiome's one of its roles can be a sort of detoxifying one, because it can metabolize things that would otherwise cause us harm. But there's been plenty of chance in the mouth and the stomach and the small intestine for harm to be done before it gets there. So it's not a first line of defense. So
No.
again, John, interesting question. And it's one of those things that seems unlikely because of the multiple active biochemical regions before you get into the gut microbiome.
Yes.
Okay, so I think we will call it a halt there. So if you can move on to the next slide, Bernadine, I'll just introduce the next speaker. So the next Coffee Science seminar is in 2 weeks' time, September 21st. And so Karen Massel will talk to us about the really intriguing, we know it's an important area, but the important topic area of gene editing for cereal crop improvement. So very much look forward to hearing that from Karen. And in the meantime, thank you so much again, Bernadine, and thank you to everyone for attending today and for the interesting questions that you brought up. Have a good day and see you at the next Coffee Science seminar.
Thanks very much. Bye.
Okay, bye.